much, and thank you for the invitation. As mentioned, I'm working at the Nestlé Institute of Health Science, and today I'm going to talk to you about the toddler nutrition and brain development. So first, I'll just do a quick overview of the brain development, and I'll focus on myelination, and you will understand later in the talk why we focus on that. And then I will talk much more about the impact of nutrition on brain development from infancy to toddlerhood. So when we're born with very limited skills or competencies, as was clearly explained on the oral motor processing capacities, but also on the motor, on the cognitive. Then we grow up and we start to have more functional outcomes, and finally we become young adults with very higher cognitive function. Now this development occurs both at the structural and at the functional level. If we look at the functional level, we can split the development in three main categories: the sensory motor, the social emotional, and the cognitive language. And as you can see here, these changes occur at different times, but in parallel. And this is the complexity of the neurodevelopment, that there are several domains that co-evolve and interact on each other. If we look at the structure, we can look at multiple neurodevelopmental processes, and I will just focus on 4 of them, but there are more. And I will explain why I focus on these 4, because they represent 2 different dynamics. You can see here the evolution across the life from birth until death of these different processes: the myelin content, the synapse density, the glucose consumption, and the cerebral blood flow. You can see they all tend to have a fast increase in early life, and then depending on the process, the dynamic is different. If you look at the early life, you can see that the myelination one is going up and quite stable up to adulthood, and then going down in aging, while all 3 others have this very fast increase in early life and fast decrease still in the early life period. And this is the reason why we decided to focus on myelination, because it is a unidirectional progress in early life, which is making our life easier as a Nestlé to find a solution to support a physiological process. So myelination, here you have the complex definition on the top. Basically, it's like the insulation cable around any electrical appliance cable going to the electricity outlet. It's just made to protect the signal and ensure that it's going along the cable properly. Additional roles that have been identified since the initial discovery of myelin is that it's also supporting the neuronal metabolism, and it's allowing a certain form of plasticity in addition to the neuronal plasticity. Now, one big problem we had to study myelination was what method do we have to measure it? And since about 10 years ago, when we collaborated with Professor Dioni in Brown University at the time, where he was a physicist, and he identified a new MRI sequence that allowed to quantify myelination. Now, traditionally at that time, we would use fractional anisotropy, and you can see here the data from 3 to 24 months. You can see a progression of the signal, an increase that is getting stronger and stronger, but it is not very clear. Whereas with the myelin water fraction methods, you can see that the signal difference across all of this stage is much clearer. And this allows us to quantify much better myelination with a non-invasive tool that is the MRI. And here on the right-hand side, you can see that this is a typical myelination trajectory that is seen through infancy up to toddlerhood. And you can see it can be decomposed in 3 main phases: an initial fast accumulation, followed by an intermediate point of inflection period, and a slower later phase of myelination. Now, why is myelination so important? Because it has been associated with cognition in early life. In this study, performed also by Professor Dioni, they recruited 257 children. They evaluated them for their myelination using this new myelin water fraction method using MRI, and their Bailey outcome to have a cognitive outcome. And what they've done then is they've correlated the myelination in the brain with the cognitive outcome. And here on the top, you can see the brain region where myelination was significantly correlated with cognitive outcomes. Now, that was trying to link the myelination to cognition what is interesting is they also did the reverse thinking process, saying, okay, we also have the cognition, so let's split the infants with above average, average, or below average in terms of cognitive outcomes, and let's look at their myelination curve. And this is what they observe, and this is what was really interesting, is that the above average are in blue, and you can see they stand out above the other infants in terms of their myelination. So whether you look at it from the cognition to the myelination or from the myelination to the cognition, it seems there is a clear association between these two. So following that, we performed a study where we investigated the impact in infancy of a pro-myelination blend on myelination and cognition and behavior. And I'll not go in detail into that because that's for infancy feeding, but in a nutshell, we gave a blend of nutrients that were supporting myelination in infancy during 1 year of life, and we measured myelination from the recruitment up to 2 years of age. And what we observed was an increase of myelination that lasted up to 2 years of age. The intervention stopped at 1 year, where we had already 20% increase, but the 20% increase was still visible at 2 years of age. Now, when we had these results, we had the same thought that many people ask us when we presented them, is like, can you use the same nutrients after 1 year of age? And if you go back to this typical myelination trajectory, so what we've shown is that from 0 to 1 year of age, we can have a nutritional intervention that impacts positively on the early brain or the early phase of fast myelin accumulation. The big question was, can we have similar or other nutrients that can impact the later stage? So just few facts about the toddler brain, because we all know the first 1,000 days, the importance of the infancy stage in terms of brain development, the fact that a lot of the brain is built in the first year of life. But if you look at 3 years of age, there is about 10 to the 12, so trillions of synapses in the brain, which are the connection between the neurons. The brain reaches about 90% of the size of its adult size. So it means like it's 90% done in terms of growth. And it's still consuming about 60% of the energy. I think it was presented earlier also by Karen, that the brain is a very energy-hungry organ. And basically, the body is spending more than half its energy to feed it during the growth period. This number goes a bit lower later in life, but still, it is a significant amount. So to investigate the link between nutrients and myelination in toddlerhood, we rely on a cohort that is ongoing in the US. It's the ECHO cohort, where they're following children from birth up to school age, and they're measuring multiple outcomes, among which dietary intake using 24-hour recalls and myelination using the myelination water fraction in an MRI scan. The beauty of this cohort is it has multiple infants that have repeated measurements of these two outcomes, which allow us to, from the dietary recall, extract the nutrient intake, and from the MRI, extract the myelination data. Now, because this is a cohort and not a clinical trial design, we had to process the information we obtained. And this is a bit complicated, so I go a bit more in detail on that, because the original correlation matrix we have here is a sliding windows where we take 5 subsequent MRI scans and we correlate them with dietary intake. And then we move them further along the age. And however, it was not really digestible in that stage, so we wanted to detect, is there time windows where the dietary intake or the nutrients that are associated with myelination are different? And this is the edge detection method we applied in the middle here. And you can see that there is clearly different patterns of colors, which are the correlation strengths of specific nutrients with myelination, which turned out to be these 3 windows. And the beauty of it is if you look at the myelination trajectory, typical myelination trajectory you can see on the right side here, these windows, which goes from 6 months to 20, 20 months to 30 months, and 30 months to 60 months, correlate quite well with the early phase of myelination, which is this very fast accumulation, the point of inflection period, and the slower later phase of myelination. And now you can see already from the correlation matrix here, there is a clear difference in the nutrients that are associated with myelination, the yellow being the nutrient associated, the blue non-associated. And you can see the first window has the highest association of nutrients. And we are still in this extremely fast accumulation of myelin. So it is a period where a lot of nutrients are important. Now, this is just a list of nutrients that were associated. There is two key messages on this slide. The first one is the amount of nutrients that were correlated on the top. You can see it goes from 60 during the fast accumulation to 20 during the inflection point to 37 during the slow accumulation. So quite a difference in terms of variety of nutrients that seems to be playing a role in myelination. And highlighted in green on the slides are the phospholipids, because we've identified them already in infancy as being very important for myelination, and we saw the similar importance as they are present in all 3 time windows. Now, this was a work done on general brain myelination. However, we wanted to see whether we could detect specific nutrient differences when we looked at brain areas that are mediating specific function. So we correlated the myelination of the brain with either language, motor skills, or vision, and we generated these masks that are the brain areas that are significantly associated with either of these functions in a baby and with myelination. We then did the same analysis of correlation of the myelination in this region specifically with the nutrients, and this is what we observe. So there is 2 things that are important. You can see that they are relatively similar, which is to be expected. They all show this very high correlation in the first window, which is the period of extremely fast accumulation, but there are some differences between them. And each of these functional brain region have sort of an individual signature of specific nutrients. We're still digesting the data and we're still processing it, but we've identified that some of them show this pattern where they are visible across all 3 of them, like the phospholipids. Others, like the amino acids, have specific windows where they are more related to the myelination. So, take-home message, I think the brain is a beautiful but complex organ, and its maturation is extremely challenging to study. I've shown you evidence that nutrition can impact it in infancy, and some association data showing that it is associated with brain maturation later in age. We have an RCT showing that in infancy we can modulate myelination with nutrition, and I've shown you the association data we've generated in a cohort, allowing us to extrapolate that such a nutritional influence in early life can also be seen in toddlerhoods and potentially later in life. And I want to thank you very much for your attention.